Hi, my name is Dr. Sarah Gaffen, and I'm a professor of immunology in the Department of Medicine and part of the PMI program. And I'm happy to take you on a little tour of my lab. My lab studies everything to do with the cytokine IL-17. So this is our favorite street corner in Pittsburgh, Sarah and 17th Street, with of course the Pittsburgh Steelers in the upper right. So we study the cytokine IL-17 and the cells that make it, known as Th17 cells. And specifically, we look at two sides of the coin of host immunity. One is what IL-17 does to protect us, and the other is how it can be pathogenic, particularly in autoimmune disease. This is my wonderful lab that does some of the work, and hopefully you might consider joining us. So why is IL-17 cool? Well, it's made by a fairly restricted subset of cells in the immune system. They're all lymphocytes, but they can be both traditional T cells like CD4 T cells known as Th17, or innate type T cells, for example, gamma delta T cells and others. The IL-17 they make binds to receptors that are found mostly on non-hematopoietic cells, so cells of the tissues like epithelial cells and fibroblasts. So in our lab, we actually tend to study these types of cells more than we study lymphocytes. And we look, as I said, at what IL-17 evolved to do, namely to protect the host against infections. And in particular, one of the things we discovered was that IL-17 is particularly important in mediating immunity to mucosal fungal infections. And so we focus a lot on oral mucosal immunity, which is a very understudied area. Conversely, as I said, IL-17 can promote inflammatory pathology in autoimmune disease, and antibodies that neutralize IL-17 were found to be remarkably effective in psoriasis and are being explored in other conditions as well. And we're trying to understand the molecular basis for this. So I'm going to tell you a short story about somebody in my lab. Rachel Bailey was an undergraduate who worked in my lab during the summer, like probably many of you. And she then came to my lab for a year to do a post-bac experience. And this is really actually about Rachel's mom. So her mom had lifelong psoriasis and psoriatic arthritis. And unfortunately, it was never well controlled until the anti-IL-17 drugs came out, which were approved in 2015. And they worked wonderfully for her. Her plaques cleared and things were great. And I wish I could stop the story there, but unfortunately, she began to experience oral candidiasis, so oral fungal infections, which might have been predicted. But even more seriously, she developed inflammatory bowel disease. And this reflected what we now understand to be IL-17's role in wound healing and tissue repair. And so when it's blocked, it can cause autoimmunity. And so this really illustrates in a single individual some of the various aspects of IL-17 function. So what does the Gaffen Lab do? Well, as I said, we look at IL-17 and fungal immunity. And so these are great projects for people who are interested in trying to understand the microbe-host interface. And we focus in particular on the oral mucosa. So we showed a number of years ago that the IL-17 receptor is essential for immunity to candidiasis. And a couple of years after that, other groups demonstrated in rare humans with mutations in this receptor that oral candidiasis is in fact one of the main diseases these people get. So we're trying to understand really the deep molecular mechanisms of how that happens. We do this using mouse models, mutants of both the host and the bug itself, and various immunological approaches. On the flip side, as I said, IL-17 can promote autoimmunity, and antibodies against IL-17 are quite effective. So we're trying to understand, the deep molecular basis for how this happens, and knowing that, could we harness it potentially for clinical benefit? Again, we use a lot of mouse models of autoimmune diseases. Some of them are listed here, so psoriasis, multiple sclerosis, and more recently we've been doing a lot of autoimmune kidney disease. And we do molecular studies in various disease-relevant cell types to try and define in a very biochemical and molecular way how the signaling by this cytokine is occurring. And that really brings me to the last part of the lab, which is defining the downstream molecular signals. This is a very complicated picture. It is, of course, just a hypothesis. And here's the reference. But we've been looking at many of the adapters that bind to the receptor, particularly the downstream gene regulation, not only at the DNA level, but also the RNA level. And regulation of post-transcriptional events has become a big focus of this part of the group. So some frequently asked questions about the lab. How many students have I trained? Well, 12 have graduated and 2 more are well on the way. So you could become numbers 15, 16, 17. This is where they are. So about half of them are faculty in various universities, some teaching, some research, some combinations. 2 of them are currently postdocs, 4 are in industry, and 1 of them left science. Sorry, that should say left science. So what's special about Pittsburgh? You're probably looking at a lot of different places. The science here is amazing. The colleagues are really special. This is a nice place to work. It's high caliber, absolute excellence, but yet it is a very hospitable place. The resources are fantastic. Your only limit really is what you can think up to do. And the city is a wonderfully vibrant city, but it's small enough to be very accessible, and it's also very affordable. And as a graduate student, that matters. Our graduate program really cares about the students. We take very good care of you. Again, we hold you to high standards, but we help you live up to those standards. And finally, if you want more information about my lab, check us out at www.gaffenlab.pitt.edu. And good luck in making your decision.